So
Okay.
um, I will starting with a question and then you guys will come up later, please.
Can you
Um,
see my screen? Yes.
yes, I can see. So what is the difference between the Rotocon from the Backbio full length 16S and the Kines full length 16S? Um, they
Mm.
two different, totally different two Rotocon are the same.
Yeah, so for the difference between the PacBio full length 16S and Kinex full length 16S is there's an additional Kinex protocol on the full length 16S part, where we actually concatenate the 16S genes. We concatenate like 12 different 16S genes in one single read, so that we can actually increase the throughput, which I will show you here. There, see this one. So this is the 16S Kinex 16S protocol. So for the normal standard monomer protocol, right, what we do is we will just amplify the 16S region and then we will sequence directly. But as I mentioned, right, the 16S is only 1.6 kb long, right? So if
Mm-hmm.
we put it into PacBio sequencing, we are actually wasting a lot of the the things that we can sequence. So what this Kinex do is we actually combine multiple 16S fragments into one single long fragment. So we combine, I think, 12 different 16S genes, I mean 16S fragments together
Mm-hmm.
into form one long read, and then we will sequence it through. So by doing that, we are able to actually increase our throughput and lower our costs.
Okay. So it means that we
Mm.
save a lot of money when we use the Kines to using the standard protocol.
No. So because you are spending a little bit of money on the Kinex kit, but it
Mm-hmm.
combine the reads together, and then you are able to multiplex more samples, and then you will be
Mm-hmm.
able to save on the smart cell itself.
Okay. Okay. But we have to pay for the kit of Kines, is it? For library
Correct.
preparation.
Correct.
So it means that if you have a trade-off between them, yeah. Okay.
Mm,
So and then my second question concerning about the equipment required for doing long read because
mm.
like in the extraction or in the QC for the long fragment size ready for the long read sequencing. Do we have to does it require any equipment specific to repair library for the long read back by all?
Yep. So we will recommend for QC instruments, for example, like the femtopulse, as well as the qubit, as well as the bioanalyzer. So we for our PacBio sequencing, we always say that if you don't do proper QC, right? If you don't you know do a QC check to make sure that your library is built properly, or your extracted DNA is high molecular weight itself, right? So if we always say rubbish in, rubbish out. So if let's say your DNA that are extracted is not high molecular weight, and then you don't do a QC, right, and then you go through library prep, and then library prep will cost, library prep reagents will cost, right, and then smart cells will cost as well. So we want to make sure you actually QC your samples properly, so that before you move on the next stage, so that you don't waste the reagents itself. So you will need femtopulse to check on high fragmented DNA, qubit to check on the concentration of the DNA itself, and then nanodrop to look into the A260 A280 ratio, to look at the contamination itself. Mm.
Okay. So do we have any special treatment to keep the fragment size in longer for to do the Shotgun metagenomic? Do we need any special treatment for the DNA to make it longer, the
Mm.
fragment size?
Yeah, I understand what you are saying.
Mm-hmm.
Just that I'm thinking how to answer this, because I think the issue with shotgun metagenomics is on the sample type itself, right? Because we know that the samples can be coming from soil, or like coming from you know blood, or something else. So in of course in PacBio itself, we do have our nanobind extraction kit, which is suitable to actually, extract high molecular weight DNA from things like blood as well as insect and, from human samples. And yeah, but I think for soil samples, things like that, our nanobite kit does not have the protocol yet, but I think there are kits from like Zymo or that is suitable for extracting those difficult to extract samples. Hmm.
Okay, okay. So have one question from Thảo Cao. She asked that E16S full length backbone sequencing sufficient for the strand level classification because she tries
Hmm.
one data from NCBI. The data come from the 16S high-fi backbone. However,
Hmm.
the data only achieve the uh, spacey, but
Species level.
yes,
Yeah.
spacey level only. Yeah.
Yeah.
But however, the spacey level also very good, but she concerning
Hmm.
the strand, the subspacing. Yeah.
Yeah, I think some of them will achieve species level and some of them will achieve strain level. So not all can achieve strain level depending on the specific whether the 16S whether the difference in nucleotide on the 16 for the particular bacteria itself, whether is it on identifiable from the 16S itself or it's maybe on the other region. So maybe 16S is not sufficient enough to look at strain level. Hmm. Yeah,
Okay.
so also not all can go under strain level. So we
Mhm,
we
mhm.
just say, compared to short read, short read usually just give you on a genus level, but for long read, we can achieve, species or strain level. Yeah.
Yeah, yeah. And then have one more question about Huy Hà. He asking, does the time taken for library preparation step is different between different kind of sample, like soil, like water, like blood, like
Hmm.
wastewater?
Hmm. I think the difference will come from the extraction part.
Okay.
The extraction protocol will be probably different, but once you get, high molecular weight DNA, the subsequent step will be the same. Hmm.
And then I'm so curious about the timing between the standard for full length and then the standard, the Kinex for full length 16S. The time preparation is different or not?
Yes, definitely there's some difference. So the process, of doing Kinex, right? There's I think around a 2 days or 2 to 3 days workflow to actually do the Kinex workflow. Yeah, but because for 16S itself, it's very easy, right? You just amplify this particular 16S region, you have the 16S primers to actually amplify this, and then you can actually do the library prep to stick the smart belt adapters onto it and run peg biosequencing. But for Kinex, because there's a concatenation process that is required, so there's an additional like 2 days workflow that is needed. Hmm.
Okay, thank you. And then
Uh,
have
I have question for this
please.
slide. How many symbol I can navigate the 16S DNA?
So in one Kinex protocol, we will like get 12 16S fragments onto one long read.
And I can ligation the 16S in meta symbol. I just I just ligation 16S on I just use Kinect 16S on in some kind of symbol like pill culture. I mean that the bacterial is isolated
Hmm.
from the mixture.
Uh, I think the like if you like the how to decide whether you want to do Kinex or not. It depends on the throughput. So it doesn't depends on the sample type that you have, but it depends on the throughput that you have. So if you have a Revio, if you're running less than 384 samples per smart cell, we will recommend you to just do the standard monomer full length 16S. But if let's say have more samples, like more than 384 samples on the Revio, then we will advise you to use Kinex. Then it will be more cost effective on Kinex. If it's lesser than 384, right, running Kinex might be a bit wasteful. Yeah, you might have too many reads for one sample.
Thank you.
Can I please have a question?
Yes.
Uh, do you treat different types of bacteria different way? For example, gram-negative and gram-positive, differently when you do the DNA extraction. That's the first thing. And the second question is that how do you know in the mix of culture and a mix of bacteria that going through metagenomic that you already sequence every single one? Like for example, you have stool sample or you have any kinds of mixed bacteria, and you know that you detect every single one in there. Thank you.
Hmm. So I think for for like whether you are saying whether how confident we are that we sequence everything that are already in the fecal sample itself, right?
Yeah,
So I think this depends on the number of reads that you actually put in. So I think the more reads that you actually put in, for example, for 16S sequencing, you are able to uh, look into more in depth of the diversity itself. But if let's say 16S is not enough to look into all the different species of bacteria in the sample itself, then you might want to look into doing shotgun metagenome itself. Shotgun metagenome will of course give you more information compared to 16S. So it's not only bacteria, but all the other things as well. Yep.
It's okay, Han. On
yeah.
good.
Thank you.
Okay, perfect.
Actually, I was asking about
Extraction.
do we need to treat the different types of bacteria differently when we do the DNA extraction? For example, uh, gram negative, gram positive, because I know that gram positive have a uh, the um, the word cell is more difficult to
Hmm.
extract. So do you have different kits for different uh kind of sample like that?
We don't. I mean, PacBio don't, but I think our compatible partners should have like this kind of like extraction kit that are suitable for metagenomics lah. I
Yeah,
think like from Zymo, they should have kits that are suitable for metagenomics extraction. Or I think Hygen should have too. Yeah.
thank you.
So, and then I have one question from Hoàng. So, he asked, what are the advantage of using PacBio for meta assembly, and what are the disadvantage that we should consider when doing the meta genome assembly?
I thought my presentation is all talking about that. No, but it's yeah. So if you're using PacBio Hi-Fi to actually do metagenome assembly, right? Of course, as I mentioned, with PacBio Hi-Fi, because our reads are long and accurate, right? You are able to get all this high quality max. All this closed genome. So when you do the metagenome assembled genomes, it's all you can see it's like you have closed genome and lesser gaps compared to when you are running short read sequencing itself. So for short read sequencing, there are regions that are difficult to sequence. There are and they will have a lot of gaps on their max itself. And I think I shown the publications, right? If you are using PacBio uh, hi-fi reads to actually do metagenome assembly, we usually see our technology actually generate more max compared to even ONT or Illumina. And the most significant one was this one, like by using hi-fi sequencing, you can see that, with our, little amount of number of data, we are actually able to actually get so many max compared to, Illumina with like, 3.4 terabytes of data, and they are only getting like around like 600 max. So that's the benefit of using PacBio.
And then what should we consider when we do it? There any disadvantage or pitfall we should notice before proceed with the experiment?
I think the most challenging part, like I think a lot of questions that have been coming through for metagenome, is of course the extraction part. Like, a lot of these soil samples, or they contain a lot of inhibitors, and you have to actually retreat them properly to actually extract high molecular weight DNA lah. So I think that is the challenge that we should be having.
Okay, thank you. And then you show me some commercialized industry like the vet for evaluate the microbiome for the cat and the dog. There
Yep.
any industrial application of BacBio for microbiome?
I think a
Like
lot
in
of
food
the
industry.
yes.
Mm-hmm.
Yes. So I think we do have some customers, for example, in Taiwan, that they actually do 16S for they are using more for human microbiome studies using 16S. So they actually do like a health screening for patients that come in for health screening. They will offer them our PacBio 16S sequencing to actually look into their gut microbiome, so that they can actually provide some kind of recommendation in terms of probiotics, or diet change. And we do have labs in Thailand that actually they actually offer 16S for food security as well. So like things that they want to actually look into uh, uh, some of their manufactured food, right? So they will sometimes run 16S on our PacBio sequencer also.
So there is any CIBD for the application in Taiwan, as you said?
No. So currently all our solutions are research use only. Yeah,
Okay.
there's no IVD for it.
Okay.
Usually customers will do certain types of validation, and then they can actually launch it as a test, as a screening test usually. Yes.
Okay. Thank you. So any further question, please? So again, the distributor of the BacBio in Vietnam is from DKSH. So DKSH là nhà phân phối BacBio Việt Nam. Nếu mọi người có liên hệ gì thì có Trâm ở đây ha. Trâm ơi, em đánh vào cái địa chỉ contact ha Trâm ha. Thanks a lot, Ting Ting. Very nice talk. Yeah. So I will starting with my first question. So in the 16S highlight, the highlight can use for the kinase or the standard protocol. They're no different. We have to adjust something.
Well,
I mean,
so
like
you have to.
if I sequencing from the library from kinase, or I sequencing from the standard protocol as Lin,
Yeah,
right?
I got your question.
Okay.
No, you don't have to. You don't have to adjust anything. The Kinect sample, you just need to resegmentation step to break the long Kinect reads into short 16S, and then you can input those 16S like standard one into this pipeline.
Yeah.
You have to do one extra step.
Okay. Thank you. So and then my second question, so do you suggest us to using the Meta MDBG rather than the HiFi ASM?
ASM Meta.
Yes.
So from our comparison study, the two assemblers are actually they're comparable. The results from them are comparable. Sometimes HiFi ASM Meta is better. Sometimes Meta MDBG is better. So depending on the different sample types. So
Okay.
I think our latest suggestion is you can use both. Both max assembled from those assemblers, and then you can feed those max and the context into our HiFi Mac pipeline for the post-processing.
Okay. So and then what is the input requirement for the HiFi Mac pipeline? Because like, do we need to sequencing with how density? I mean, the depth of coverage for the HiFi Mac. Do we need very depth or because as you show in your talk, you have very different kind of coverage.
The input is just the output from your assemblers, but if you want to recover more max, definitely you need higher coverage of your data.
Yes. So I just want to asking that how much is it? How
I
depth?
don't know. Depend on the diversity of your species in your sample, right? So like in our study for the human fecal reference, the Zymo one, they just have 600 species in that sample, and we use just the 4 SMRT cells to reach the saturation. But for the soil samples, we use the 4, but and you can see that the number of max is still increasing.
Okay. Okay. So it means like
Yeah,
acceptable to sequencing. We don't need to do the mark, but at least we can binding them. No. But at least we can build up the context for them. So the HiFi Mark highlight can stop, no, not stop, like just limit to the context um, generate the context only. We don't need to go to binding and to closing the genome.
Like the HiFi Mac pipeline, the input of that pipeline is the max, the maybe the Cmax, the high quality max, and also those linear context. And the HiFi Mac pipeline will help you to further binning, like to assign those linear context to whichever species
Yeah.
belong to which max.
Yeah.
Yeah.
Because like I asking because like we sequencing not really that, and it means that we cannot go further to binding and then finish closing the genome. So the pipeline can help us to stop early, like the context only in the context step or not, or we have to do the full of the pipeline, the full of the whole pipeline.
Mm.
Did you get what I'm asking? Sorry that I'm confusing you. Okay, so move to the question from Sir, please. Can you please directly asking Sir?
So in a scenario with limited memory capacity, so which method or best practice type would you recommend for metagenomics profiling? SORMAS, Diamond Megan, or Minimap Megan?
Oh, I don't have a quick answer for your question. Yeah.
Yeah, because it's very memory consume a lot. So and then we don't have
Yeah, that's true.
the big HPC over here. We only have the small station. So then I think that the sound map is the best because I try with the sound map and it look okay. Yeah. And the sound map is not require a lot of, especially we do with the camera around 21, camera
Yeah.
21 or 35 is okay. Yeah.
Yeah, I agree. Because the diamond and Megan is based on the protein alignment, so maybe it will consume more memories.
Yeah. Okay. So are there any more questions, please? It's a very good opportunity here. We have expert Ting and also Ray come from BacBio, and they know everything about their pipeline and so on. So please taking this opportunity.
Yeah, Yeah, I mean, if you have further questions, you may also pass your question to DK Stage, and
Mhm.
they can reach out to us.
Okay,
Yeah.
perfect. Okay, thank you, Ray, thank you, Tinh Tinh, thank you, Trump from DK Second Back Bio. So now we move on to the next platform, the ONT. Thank you a lot for your time. Thank you.
Thank
Thank
you.
you.